So, I have the pleasure to welcome our speaker this evening, Tom Bruns. He's been a professor at the University of California for a long time and has been a great contributor to the society and has spoken for us on a number of occasions, both at the general meetings, at the Fungus Fair, and other events. So it's great to welcome him back in his talk tonight on forest fire and fungi, losers, winners, and strategies in the positive or post-fire environment. Sorry, Freudian slip there. Yeah, I'm really looking forward to this talk. As we all know, it's been an interesting spring and summer again in forest fire situations. So a little bit about Professor Bruns. Professor Bruns joined the faculty at the University of California, Berkeley in 1989, where he studied fungal ecology and systematics, and has published over 150 scientific papers in this area. He served as president of the Mycological Society of America, and is a current fellow of the American Association for the Advancement of Science, the California Academy of Sciences, and the Mycological Society of America. He has won teaching awards from the College of Natural Resources and the Mycological Society of America. So please join me in welcoming Professor Tom Bruns.
Thanks for that introduction. Is this on? Is that on? Is that close? Okay. Let's talk about fungi and fire. And of course, I send these titles out, way in advance, and then forget about them. And then when the time comes up, I come up with a better title. So this is actually the better title. Because and it'll become clear why this is the better title, and it has nothing to do with the best of fires or the worst of fires. Yeah. But it does have to do with two fires that occurred during my career that just opened up all kinds of opportunities for research. And almost everything that I'm going to tell you is based on those two. So here's what I'm going to tell you today. I'm going to go through some basics of plant fire ecology, and I do that because in some ways I'm a frustrated plant ecologist. I'm very envious of what the plant people know about their organisms, and fungi, we just never quite have that much information. And so every time I find something out with fungi, I go back into plant ecology and realize I knew that 3 decades ago, and it's just it fits in with one of their general rules. So we'll start with some of their general rules. And then I'm going to introduce you to the two fires that are the tale of two fires here, the Mount Vision fire and the recent Bruns fire. And then I'll talk about the importance of ectomycorrhizal spore banks. And if you don't remember anything else from this talk at the end, that's what I want you to remember, is that ectomycorrhizal spore banks are the main story for post-fire regeneration of the ectomycorrhizal community. And if you don't know what that is, you will by the end. Then I'll talk a little bit about how it doesn't appear that at least ectomycorrhizal fungi perpetuate themselves vegetatively between fire events, at least stand replacing fire events. And then I'll move into the area of fire-specialized saprotrophic fungi. Okay, so plant ecology. If you think about plant ecology, there's a lot of known fire strategies out there, right? That there's, especially just in the pines, you can think of things that survive ground fire by having thick bark and pruned limbs and a canopy that's way above the fire, ponderosa pine being the stellar example, but there's lots of others that do that. And they survive fire events by just being big and above it, basically, and well insulated. There's others that survive by rootstock and root sprouting, and in California we've got a ton of root sprouters. I'll show you some other pictures that give you some examples there. There's other pines that survive by having their seeds protected in the cone. The cone is actually closed until fire events, so they're very adapted to fire. And then they release the seed from the cone, and they're there to repopulate right after the fire. Lodgepole's sort of the poster child for that. Jack pine in the east but our own bishop pine, which I'll show you, does a great job of it as well. There's a number of others. And then a lot of the herbs in the tree area, aspen, can survive in the soil seed bank by putting their seeds in the soil, and they slowly get buried over time. And they can sit there decades until a fire event. When we had a big fire event in Yosemite a few years ago, there was a Clarkia that hadn't been seen for ages that showed up in the post-fire event because it was keyed to go off when the fire roared through and the seeds were released. So the other thing about fires is that they're not all a monolith. You get different types of fires, right? And so those fires can vary by what's called the fire regime. And what's the picture is how you figure out sort of what the fire return interval is in an area by the fire scars. I borrowed this slide from one of my colleagues, Scott Stevens. And the components of a fire regime would be the frequency, the season that it occurs in, the size of this fire, how big it is, the severity, meaning how much killing there is of the trees, and then the interactions between the fire and other events going on. So I'll give you two examples of fire regimes. The first is this low severity fire where it stays on the ground in the case of a fire, in the case of a forest, and generally not lethal to the overstory. It's going to kill some trees, but these are going to be little trees in there. And if it's a very hot ground fire, it could kill some bigger trees as well. And the frequency of these things is relatively high. So in California, we get these, like the mixed conifer forest, for example, had a fire return interval somewhere between 5 and 15 years. And that's what really maintained it. And those fire events traditionally were started by lightning, but also by Native Americans. So this was what we saw in the Sierras, for example. And you can do this artificially by taking a plot like this and burning off the fuel in it by picking your conditions and lighting it. And this is the kind of thing that Scott Stevens does a lot of. He was involved with some of this up at Logic Force, where we got to sample below ground before he did this and then burn through. And so this kind of a fire, low intensity fire, is something that's open to experimentation. You can look at what's there beforehand, you can burn it, and you can say, okay, what survived? How did it recolonize? And certainly with plants, that's all well known. With fungi, it's not so well known, and I'm not going to talk about it much, although there's been a fair amount of work on this now. So this is what that spot looks like by the next spring. You can barely tell it's burned in the picture. You can see some charring and so on. But that's what a low intensity fire does. It just sort of cleans things up and keeps it into a park-like open forest area. So now we can contrast that with a high intensity, high severity fire. This substantially changes the forest because it kills everything. Historically pretty uncommon in the Sierras, but as you know from reading the news, it's becoming much more common. And the reason that it's becoming much more common is in part that we've done such a great job of suppressing fire. And so by suppressing fire, we built up the fuel levels, and then when fires do get loose, they get loose under the worst possible conditions, when things are really dry and when the winds are high and so on. Then you get, with these fuel levels, you can get tremendous stand replacing fires. So this is, as I mentioned, it's not common in the Sierras, but it would be common in certain forest types, lodgepole pine, for example, that fire that took place in Yellowstone a few years ago, very typical of a lodgepole pine forest fire, in that those return about every 100 to 200 years, and they are stand replacing fires. And the reason they're stand replacing fires is lodgepole pine, mature lodgepole pine looks like that. There's fuel everywhere. There's great fuel continuity between the ground and the canopy, so it gets carried up, and then the canopy is tight too, so it carries crown fire very easily. But in lodgepole, when that happens, it looks like this a few years later. And that's because the cones, a closed cone pine opens up all these seeds, broadcasts them out, and then you have smoke from the fire coming off there. And then that's a little spot in Limiter Ridge, actually one that we studied. That's a picture of the crown fire going on, burning up bishop pine. And this is a picture of me in a much younger manifestation, going through and finding a site that we had pre-fire. And so the advantage to this fire, and the advantage to the ring fire as well, is that we had tremendous pre-fire data for an event that you cannot sort of predict in time. And so it turned out to be really useful. So back to the fire. This is the way it looked from Limiter Road as you looked across a great big burnout area, a little bit of green in the parking area. But very shortly after that, you get resprouting. There's all kinds of things resprouting coming up plant-wise. Here it is just May of the next year, and already looking pretty green. The pine, again, was a closed cone pine. This is bishop pine. You can see all those cones up in the canopy, and you can see how they've opened from the fire. All of the little brown in the lower image there are wings of pine seeds. So they were just unbelievable number of pine seedlings on the seeds on the ground. And here we are just 2 months after the fire, and there's already pine seedlings coming up. Now the little sand spot there was actually one of the soil cores that we had done before the fire. Because what we'd done is filled them with sand so that we didn't leave holes in the ground. And after the fire, they were very visible where they were. So we could go right back to those spots where we knew what the mycorrhizae was below ground of the mature trees and sample of these little seedlings later. Here are the little seedlings just 4 years after the fire. And if you've been out there now, they're almost this thick still. I mean, it's amazing how thick the pines are out in the woods. Fire 2 happened last year. This is the Forest Service map, actually their poster of the rim fire. The "You Are Here" is where the map is located. It was the third largest fire in the state of California. It covered over 1,000 square kilometers, so big fire. And we had 2 sites in the middle of it. These sites we had as part of a continental study of pine forests across North America. These are 2 sites out of 68 that we now have. And we had an extraordinary amount of pre-fire data on these. Here's what the rim fire looks like from Highway 128. And here's what our sites looked like before and after. So this is when we were, this is Kabir Pay coring the site, pre-fire, and then post-fire we're back setting it up. Here's another blow-up of that picture. You can see this is the first of the 2 sites. Every tree it was big crown fire, killed off everything, burned off all the canopy. Lots of burned out stumps like this. You get in where the fire has burned down through where the roots are, and the whole area around it looks like pottery because it was so hot. This is the second site, and on that site, the trees were killed, but the fire was a lower intensity. It went through on the ground and didn't burn out the needles on the canopy, so the needles came down later, but the trees were all dead. So these sites, we went back to look for them. Here's a happy postdoc. She's happy because she's holding a wire from the pre-fire flag that we use. So in each of these sites, we had 13 spots where we sampled the ground in various ways, the below ground fungal community in various ways. And this plus GPS plus measuring the trees and all allowed us to find each of those 13 points at both sites down to a distance of less than half a meter for all of them. So we're basically right on top of the same sites that we sampled pre-fire. Now the other difference between these 2 fires, they're both big rip-roaring fires, but one of the big differences there is the pine species, right? That when we were up in the Sierras, the pine species was ponderosa pine. This is not a pine that has closed cones, so it does not carpet the area with seedlings. We had to go and plant seedlings there methodological. When we did the Mount Vision fire, we were basically pioneering these methods, and they were primitive. I mean, now when I look back at what we did, it looks like the Pleistocene. I mean, it's amazing how simple the methods were at the time. But what we had to do was we would take a soil core, we would find mycorrhizal root tips like that one, and we'd take individual mycorrhizal root tips, extract them, and we actually would riff lip them, do this restriction digest thing first, match them, and then we'd sequence a small subset of them because sequencing them was tough at the time. And then you'd match that, and there was basically no database at the time. And we'd match that to things that we'd collected, and we could say, okay, this was Suos, for example. Now, by the time the Rin fire came along, the genomics revolution had roared through, and the methods had changed dramatically. So now, a lot of our data from the pre-fire sites of the Rin fire was soil data, where we could just take the soil and sequence everything. No more picking one root tip. You just take the whole thing and produce literally millions of sequences. And so when you do that, you sort of lose the ability to look at them all. You have to put them through these computer pipelines to visualize sort of what you saw. But you can get back to this stage after you filter through and you want to look for this genus or this set of fungi, and you can still do that. But now you can look at all the fungi. You don't have to restrict yourself to just the mycorrhizal ones, or just the ones that are colonizing on that particular area. So your sample size goes way up, and the coverage of fungi that you can look at goes way up. So the Rim fire was nice that way, is that we had this much better data. So the questions here I want to ask are, first, are there any truly fire-adapted fungal species? So there's fire-adapted plants, clearly, like the closed-cone pines. Do we have anything like that in the fungi? Do fungi have any equivalent strategies to the plants? And how do fungi recolonize after fire? So if we go back to this plant list, are there any common strategies here for fungi? I think two of these we can wipe out as unlikely right from the beginning, is that we're not going to have above ground survival of fungi, that everything is below ground there. Or maybe in the plant, like some of the woody ones, maybe you could call that above ground. I'll come back to that. But basically an unlikely strategy. Similarly, you don't really have a lot of spores up in a burnt canopy likely. So those two are probably not too likely, but we could get something like survival of mycelium below ground, and then we can have the equivalent to the seed bank as a spore bank. And that's where I want to go first. Remember, that was my one take-home message. If you don't remember anything else, you're going to remember that ectomycorrhizal spore banks are the big story. So that's where we're going to go first. That's the story we know the most about. So some of you are going, what the hell is an ectomycorrhizal anyway? Okay, here it is. So this is ectomycorrhizal interactions are these ones between the fine roots of trees like pine and fungi like Boletus, for example. And it's basically a commodity exchange where the fungus gets carbohydrates from the host, and it gives nutrients to the tree in exchange for it. And the one thing that's really important to know here is that it's an obligate relationship for both. That is, you cannot grow pine trees without ectomycorrhizal fungi, and you cannot grow ectomycorrhizal fungi without the trees. So it's kind of a chicken-egg thing, and after a fire and you've killed off the main host, what's the fate of the fungi, right? And how do the pines get back without the fungi? Okay, something else you're going to have to know to follow the rest of this talk is how to read one of these diagrams. This is what's called a ranked abundance curve. And what this shows, each one of those little columns would be the abundance of a different fungal species in ranked order. So on the far left would be the most abundant, and on the far right would be the least abundant. And this is a very typical way to display the community structure of different types of communities, plant communities or animal communities or fungal communities in this case. And ectomycorrhizal fungal communities tend to be like this in that they are hyper-diverse. There are no true dominants. That if you look at the most abundant species on this curve, and you don't know who those are because I haven't given you names here, they'd only account each for maybe about 7% of the total abundance. And then there's this long tail of rare types. And this graph that I'm showing you is just a piece of that other graph that's above and inserted. And this came from, I think, a 4-hectare study of a mixed conifer forest in Sierra National Forest. So huge numbers of species, hyper-diverse. And you guys that go out collecting, you know this, right? You see, I mean, when Mendocino, when you come in and lay out all the fungi on the table, this is why. Because there's so many fungi below ground. But when you look below ground, as opposed to picking the mushrooms, it's even worse. Because you get them all. You get the ones that aren't fruity, or the ones that you would never pick up, like all those nasty little anosmias. Could say that since Brandon's not here. So let's return to Point Reyes. So prior to the fire at Point Reyes, we had done, using rift lift and the primitive methods of the time, we had looked at 3 different sites at Point Reyes, and here I've summarized those sites by rank abundance curve at the family level. And what you can see is that the Russulaceae, the Thelepheraceae, and the Amanitaceae are the big winners here. They are the ones that pre-fire dominated that forest. And then there's a long tail, but not a particularly long tail because, again, our methods are primitive here. We cannot extend that tail very far because we can't poke enough holes in the ground or hire enough people to amplify the root tips at this game. But those were the things that dominated. After the fire, the Russulaceae, the Thelepheraceae, and Amanitaceae are basically not represented. And those same 3 sites look like this. Still, you've got a ranked abundance curve. The little asterisks above all of those are all rhizopogons. So rhizopogons win. And the rest of them are mostly ascomycetes that weren't there before the fire, at least on the dominant part. Maybe they were way out on the curve and we didn't see them, but post-fire they were there in huge numbers. So I don't know much about who these were yet, even for the Point Reyes fire. But I do know about the rhizopogons, so let's talk about rhizopogons. There are 5 things to know about rhizopogons. They are all host-specific. So like rhizopogon at Point Reyes, it's either on pine or it's on Douglas fir, it's not both. And there's a relatively small number of species out there. As you get to the Sierras, where we'll go next, there's many more species, but they still are host-specific. The fruit bodies look like these little truffle-like things, and you have to find these below ground. That's where they fruit. And if you're a rodent, you can find them very easily. And if you were a human, a mere human, I have great trouble finding them, but if you were Jim Trappy, you're almost as good as a rodent. You can find them all over the place. They are not usually the dominant colonizers of the mature trees, but they're really common on young forests. And they have these long-lived spore banks. That's what you just saw in that last slide. What allows those to colonize all those seedlings is that the forest soil is just loaded with rhizopogon spores. We've tried to sort of estimate how many spores per cubic centimeter of soil. I've never come up with a good number, but I'm convinced that when you walk through a pine forest, there's really no soil there at all. It's all rhizopogons. Because it's just so dense. You can dilute forest soil something like 100-fold. Any pine forest soil in California, you can dilute 100-fold, and still over half the seedlings will come up with rhizopogons. So there's a lot of spores out there. And they germinate readily when they see uncolonized plants. Here's one of those sorts of experiments. So this was at Point Reyes, pre-fire. And what you're seeing is what was on the mature those same soil cores, this was a little transect that we did with groups of 3 soil cores. On the same soil cores, we just took the soil and dried it, removed the roots, and then planted them in little pots and put seedlings in it. And then looked at what colonized those seedlings. And then that's the list on the right-hand side. And all of the blues are rhizopogons. So you can see those blue aren't on the left-hand side, right? But they are on the right-hand. So it's the same soil. But when you're looking at the active trees, the big trees, they're not interacting with this rhizopogon spore bank, with the seedlings, when it's highly disturbed, will. So it's just like a seed bank, right? But it's a spore bank. And that's what the root tip of rhizopogon looks like. So this is what happened at the Point Reyes fire. The mature forest looked like that at this one site. The bioassays looked like that below. You can see all the rhizopogons. You can see that one was present in the mature forest that we picked up that turned out to be rhizopogon salabrosus, and it did well in bioassays. But there's a whole set that we didn't see in the mature forest at all, but they show up in the bioassays. Here's another one of these where we're looking at the pre-forest bioassay and the pre-fire bioassay and the post-fire bioassay from the same site. You can see that they look pretty similar. There's a few things that show up more commonly after the fire, though, and one of those is this, Rhizopogon alvasiotactus. This is one that is going to be a recurrent theme of this talk because it showed up again and again under these sorts of circumstances. So here it looks like it increases post-fire on the bioassays. When we look at the, so this is post-fire bioassay on the bottom, and these are post-fire seedlings, so the ones that are actually out there on the ground at the top. And you can see they look a little different because you pick up some additional things that come in by wind, like the hubuloma. But the rhizopogon still holds its own, and the biggest rhizopogon there is the alvasiotactus right here. So let's go to the rim fire and look at the spore bank, and we're going to see the same thing. This is a ranked abundance curve of the pre-fire bioassays and the post-fire bioassays. In this case, they're stacked on top of each other. So you can see the first one is a rhizopogon, and it shows up both pre-fire and post-fire bioassays at about equal frequency. There's a few that are not equal. For example, Cenococcum and Suillus, they were common in the pre-fire soil. Post-fire, they are gone. And here's Rhizopogon alvasiotactus, and it did the reverse. It was there pre-fire, but post-fire, it's about twice as abundant as it was pre-fire. Now this side of the curve is hard to interpret because when you get this big long curve, these rare types, you're barely sampling, and so when it shows up either pre or post and not in the other category, it could just be a statistical quirk, and so there's no statistical power to sort these guys out. But if we look at them, there's some evidence that very similar things are going on. So for example, Cenococcum, one species of Cenococcum that disappeared, that was pretty abundant pre-fire, didn't show up post-fire, and the only other 2 representatives we had that were rare also didn't show up post-fire. Similarly, the Suillus that didn't show up post-fire, the abundant Suillus, the Pseudobrevipes, was joined by 2 other species of Suillus that were rarer, but also were not seen after the fire. And the reverse is true, that if we look at the rhizopogon here, the Rhizopogon alvasiotactus that increased, there were a whole bunch of other rhizopogons that were common both pre and post fire in the abundant things. And when we look at the rare ones, the majority of the rhizopogons show up in the fire column and not in the unfire column. So even these rare ones, I think we're seeing some real signal there, and there's a patterning here to who wins and who loses in these settings. So Rhizopogon alvasiotactus again. So let's talk about this in more depth. What does this thing look like? This is the one and only fresh collection I've ever seen. Actually, it's not quite true. I've seen this same collection a couple years in a row would go back to the exact same spot and find it. There's one place on Mount Vision where I've seen it. How many people in this room have seen Rhizopogon alvasiothincus? Really? I'm impressed. Because it actually was listed as a potentially rare species because it was known from only 2 locations. But it turns out that when we started surveying pine spore banks across the state, it was everywhere. It's one of the more common species. But the catch is that I don't think it fruits very often. So this was a study that Antonio Izzo in my lab did a few years ago up in Sierra National Forest. And what he's showing here is a ranked abundance curve of the bioassay seedlings. All of the little starred ones are Rhizopogon species. And all of the ones with the little green dot over them with a percentage are the ones that were also found in the mature trees in that same forest. So you can see most of them aren't found in the mature trees, or if they are, they're low abundance. And in fact, the only Rhizopogon that was found at all was the Rhizopogon salabrosa at 1% in the mature forest. All the rest were only found in the bioassay. Now, that doesn't mean that they're not out there again, because you've got this rare problem of finding rare things when you're poking holes in the ground and looking at roots. So we employed the squirrels to go find them for us, and we did that by looking at their scat. And so you can amplify the scat and say, okay, does it have this Rhizopogon in there? So when he did that, that's what the little squirrel icon is showing, which ones are out there and the squirrels could find. So there's some of those that, you know, must be on the roots somewhere, but they were rare enough we weren't finding them, but they were fruiting and the squirrels found them. But there are 2 that we didn't find and the squirrels didn't find, but were out there in the bioassay to be found as spores. And those 2 included Rhizopogon alvasiothincus. The other one was Oxentalis, which is another very rural species. Now the same soil, if you heat it up and then plant the bioassay into it, you can find out, okay, you can ask the question, are these things resistant to heating? And this, again, was Antonio's work. And what I'm showing there is that what the heating profile of the soil looked like. He made an effort to try to make it look like a fire where the heat goes up very quickly and then tapers off slowly. And so he did 3 different temperatures, 75, 55, and 45 degrees, and then planted seedlings into it and looked at what that did. And this is what he saw, was that as you go up to 75, you get a slow shift in what's there. And you can see in particular that the Rhizopogon alvasiothincus increases as you heat it. So we don't know whether it's being stimulated by this or whether it's just now colonizing more of the seedlings as other things are killed off. But you can see some others have killed off. For example, the Sinacocca, which we saw in that other one also dies off when it's heated in experimental conditions. Yeah, that's that. And so then Kabir Pei repeated a very similar experiment where he looked at the effect of ash and heating on soil, and now he's using point-raised soil. And so he either heats it or adds ash or does both or does neither in all possible combinations. And this is a slightly obscure diagram that I'll explain here. Each of those little spots would be the community of fungi, represents the community of fungi on a set of seedlings under each of those conditions. And so if 2 spots are near each other, they share more fungi in common. So you can see, for example, here, that all of the little gray dots that are heated dots are near each other. So they share a common mycorrhizal community. And the ones that are not heated have more variance. And on the diagram next to it, the species that responded to this are shown. So for example, here is Rhizopogon alvasiothincus, way up in the fire burn site. So it becomes more common when you heat the soil. Here is viability by a fluorescent stain. So here is Rhizopogon alvasiotectus. It's the top curve there. So it can withstand something like 4 minutes at 65 degrees and still be, most of them are still viable. Here is Lucaria proxima. After about a minute, most of the viability is gone. So it looks like there's some real differences between how these fungi behave with respect to heating. So going back to this question, are there any truly fire-adapted ectomycorrhizal fungal species? I would say that Rhizopogon alvasiotectus is a very good candidate for that. And it's because we only see it commonly in the spore bank. Spores are resistant to heat and perhaps activated by it. And then it's common on post-fire seedlings, at least in Point Reyes. And we'll know whether that's true very shortly up in Rimfire as well. So if we go back to this, we can say, okay, we've had survival of spores. So that's similar to survival of seeds in the seed bank. What about survival of rootstock? Do we have anything that survives vegetatively? Well, Point Reyes fire allowed us to look at that in the case of Pseudomonas pungens. And it allowed us to look at Pseudomonas pungens because we had one site where it was a big fruiter. We mapped all the fruiting bodies in and we genotyped them. So we could say this genotype covers a huge area. And then when that burned up and Pseudomonas pungens started fruiting there again, we could go back and re-genotype it and go, okay, did that genotype survive? And when we did that, what we found was that all of the post-fire genotypes were new. That there were no genotypes that were the same pre-fire and post-fire. So that makes it look like it's recolonized by spore and not mycelium. Now this is an N of 1, so we don't know whether other things do survive the fire. But what we do know, if you think back to those ranked abundance curves, there's a tremendous change between what is on the mature trees and what shows up on the seedlings. So the first approximation, all of the early winners are losers after the fire. They die. And if there is mycelial survival, it's rare and sporadic. It still could be very important over the length of time for the forest, but it's a rare event that's hard to capture when you're looking for it. So we can't really say it doesn't survive, but it doesn't look like it survives often. Now, so far I've been talking about mycorrhizal things, but let's turn to the saprobes a little bit. Point Reyes fire, one thing that was really obvious about a year afterwards was all the standing wood was being decayed. Now, most of these things that are decaying here that we saw are also things that you might see post or pre-fire and are good early colonizers by spore. So I think the most likely explanation for most of these is that they colonize post-fire by flying in by spore. There's a few of them though, particularly the Coniophora, which is very common below ground on roots and so on, that may well have survived as mycelium and crept up on dead wood and grew up, but we don't know yet. So going back to this question, are there any truly fire-adapted fungi? I said, well, ectomycorrhizal fungi, yes. What about saprobes? Well, with saprobes, there was already a great literature on the post-fire saprobes that we had basically ignored. And we ignored this for two reasons. One is at the time I was very obsessed by mycorrhizal fungi. And two, the saprobes were much harder to deal with because we could not pick out individual root tips and sequence them. They were in the soil. And at the time, there was really no way to look at their mycelial stage in the soil. But going back to the literature goes back to the early 1900s. And one of the classics came out in the late 60s. Still, and actually a great experimental paper that goes through with experimental burns and looked at the fruiting pattern for a whole bunch of these so-called fireplace or fire-following or phonesioid fungi. So let's look at some of those. They did show up at Point Reyes, especially Anthracobia. Anthracobia was all over the place. And then this Rhizina undulata showed up about 6 months later and was super common. disappeared now. None of these could you find at point rates now. Very short time window where they show up. And the ecology of them is very well known in terms of their fruiting. They're very much fire-following species. The rimfire was rich in these things. We saw lots of them, including some basidiomycetes, but mostly ascomycetes. And some of these were relatively easy to identify, and some of them, you really have to crawl down on the ground to find them. Here's Elsa looking into a burnt-out stump hole to find this spore mat. These things were really common in the rimfire all over the place, and when you look at them under a microscope, they're loaded with spores. These are spore mats of Pisizalian ascomycetes, and we don't know much more about them than that. We've got sequences from a few of them now, but not enough to tell you much. So here's the kind of data we had from the rimfire pre-fire. We had composition of the bulk soil community. We looked at the active community through several seasons with something called in-growth bags. We had the ectomycorrhizal spore bank community, which you already saw, and then we had the ectomycorrhizae on the roots. Let's turn and look at the bulk soil community because we've got one stellar result on that. So we were able to get back in and resample 1 month after the fire. So we were there in October, got in, collected soil, and then put it through a high throughput sequence method called Illumina sequencing that produces, I think, 20 million reads or something like that. This is what we found. 68% of all of the sequence reads were a single species. Now remember those rank abundance curves where things are usually like 7% or something? And it was dominant at all 26 sample points. So not only was it super abundant, but it was very frequent. You couldn't find a place in the forest where it was not. The second most abundant to you was another member of the same genus. And just for comparison, the three most dominant sequence reads pre-fire were Cebusina, Inospe, and Calambulina. Each one being less than 7%. Okay, guys. We're off. Pyrornema amphiloides is the hands-down winner 1 month after the fire. When we went out there, in spring looking for it, after we got this result, it was like, what the hell is that? We gotta go find it. So we went out looking for it. It actually was not an abundant fruiter. And when you go back to the Danish fireplace fungi and look up Amphilota or Pyrornema amphiloides, it is, he actually says that it doesn't cover it because it's so sporadic on the fruiting. So it's not an abundant fruiter, but it's really a dominant below-ground. Okay, so what do we know about this? We know that it fruits rapidly after fire. That's in the literature. It can show up, I guess, as fast as 2 weeks after fire, or a month. Fruiting is restricted to burnt or heated soil. You never find it anywhere else. Very rapid growth in sterile soil. This is when you're doing it on, in the laboratory. You can inoculate it on soil and it grows. Homothelic fruiter, so you don't need to mate it. One strain is enough to make the fruit bags. And I should point out, these are the fruit bags on the side. These are little tiny cups clustered together on a slightly out of focus picture. Sorry. And to our glee, the genome sequence has been completed and assembled for it. So what that means is that we've got amazing information on what its enzymatic capacity may be, and it's opened up all kinds of tools on how we can look at this. And now we know it's a post-fire dominant. Okay, what don't we know about it? actually don't know how it persists for 50 years or more. And I'm assuming it's persistence, because I can't imagine how it could have dispersed into 26 different spots and taken over the soil in a month, given that it wasn't fruiting anywhere else before that. So I'm assuming it persists, but we don't know how. What we know is that the ascospores are thin, flimsy little things. There's been evidence that they can hold out for 2 years in the laboratory, but over that time they go from about 90% viability to about 3. So it doesn't look like that's the answer on how it survives. We actually don't know what it eats. Here it is in the soil. There's a lot of things that it could be doing in the soil, and we don't know what those are. And we don't know how long it remains active in the post-fire soil, but we have those data now. I just don't have them for today. And we don't know what part of the post-fire soil it's actually in. We sort of bulked it, so we don't know if it's at the top or down lower or where it is. But we can have some predictions, and we can have some predictions on it based on what we know about what fire does to soil. And what we know about fire and soil is that there's a very steep heat gradient, and it typically does not penetrate very far because dry soil in particular is a good insulator. But what it does do is it changes the carbon up near the top by burning it or partially burning it. It takes all the wax and pushes it down into the soil where it then congeals and makes a hydrophobic layer. And in doing that, it seals off the water vapor that it has also pushed down and condensed. So you actually get a water layer slightly down in burnt soil, counterintuitively. And then below that is the area where the first organisms might survive. 70 degrees is considered sort of killing temperature. But you can figure that above that there'll be little pockets where it probably doesn't reach that temperature. So what we're expecting is that there'll be a depth gradient on where this thing is activated and where it rules the roost, and some interesting substrates that it may go for post-fire. So this is sort of got us enthusiastic for going out and lighting prescribed fires now. And so what we want to do here is we want to create some small hot campfires, basically, and monitor the soil temperature below it, and then assay fungal community at time points below that. We want to artificially heat soil in the laboratory to selected temperatures from that gradient back there, and then inoculate pyronema on it. And then we want to use the messenger RNA from pyronema to ask what genes it turns on when it sees these environments. And that is going to answer that, what is it eating? So the conclusions of the talk are that some fungi certainly have strategies for dealing with fire. Unlike plants, we really do not understand their survival and behavior very well. We're just really starting to get a grip on this. So tools really are just becoming available to dissect these systems. Invite me back in 2 years and I'll tell you more. I just want to say that people have worked on fire in my lab since the Point Reyes fire, and there's been a whole crowd of people that have helped put together these studies that I'm telling you now and taking credit for, but it was very much a set of students and postdocs that did this work. And I'm happy to answer whatever questions I can.
I have 2 questions. What have you learned about the succession population change? Have you marked that yet? And the other question is just sort of a fantasy of, could those organisms be living in some other bacterium or other living organism that's protecting them until they reach that temperature?
So the succession story is a long one, and I can't answer that very quickly, but we did follow it out at Point Reyes for a long time, and particularly in these little pine islands that established post-fire. And what we know is the community is simple for quite a while, It gets dominated by especially Suillus and Rhizopogon for the first decade or so, but by the second decade, it's starting to look more like a mature forest again. And then the second part about hiding, what I forgot to mention about the Pyronema was that we had the pre-fire soil data. It was not in it. We could not detect Pyronema in any samples, any of the 26 samples that we had from the pre-fire. We could not pick up Pyronema. So it's either really, really, really rare, which I think is what's going on, and it's just below our detection limit. We're trying it with there's a different way to go at it, and we can still have the samples, and we go back and go, okay, now we've got a specific tool for just Pyronema, and we'll go back and see whether we can pick it up. But it doesn't seem to be, if it was there pre-fire and doing something else, it was not doing it abundantly.
Okay, thank you.
What assay were you using pre-fire? What assay were you using?
So pre-fire was high throughput sequence as well.
Okay.
But it wasn't Illumina, it was pyro sequencing, 454 sequencing, which does not have the depth of Illumina. That I'm probably talking over some people and right to others.
That orange-red colored, the picture that you had with the Pyronema unfoliatus, was that the color of the mycorrhizal?
This. So Pyronema is a saprobe, it's not mycorrhizal. It's not mycorrhizal. And that's the fruit body, or clusters of fruit bodies.
Did you find morels pre-fire? I mean, in the soil?
I don't remember. It was not, they were not abundant.
No.
They were not abundant. And they weren't abundant 1 month after the fire either. I mean, the only thing that was abundant 1 month after the fire was Pyronema. Everything else, after the 2 Pyronemas, I think the next most abundant thing was less than 0.5%.
So if you didn't find a high percentage of Pyronema before the fire and so extremely abundant within a month after the fire,
Yes.
your assumption is it grew due to the heat stimulation or something extensively and gave you that much abundance within a month
Yes.
from rarity initially?
Yes. I think that how it does it is with sclerotia.
I was gonna
Because
suggest that.
supposedly there's 2 species of Pyronema. I'm a little dubious about the species borders on these things, but the domestica makes sclerotia in culture, these little micro knots of hyphae. And Amphilotes supposedly doesn't, and that's actually the only consistent difference between those 2 species. My guess is that Amphilotes does do it, it just doesn't do it in culture, and that those are what rest.
There's a place in South Africa called, part of South Africa that has an ecozone called the fungal.
Yes.
And they discovered maybe 10 or 20 years, 15 or 20 years ago that many of the trees there, it's not the heat that stimulates the seeds to sprout, it's the smoke extract that comes out. And so now you can buy seeds of horticultural varieties from South Africa that are already pre-treated with smoke extract.
Right.
And so it sounds like what you mostly did was heat testing of the species. Did you do anything with smoke extract, or do you think that
We
had
did.
any changes?
We did ash treatments a couple times, so ash has a lot of interesting leachates that often stimulate seeds as well. It has some effect, but not much. And certainly a lot of these don't need fire to stimulate them. I mean, if we pick these up in a spore bank as soon as we eliminate all the other competition. So they can respond to roots without heating. But probably not the saprobes. I mean, the saprobes I know almost nothing about in terms of what kicks them off, but we might be able to stimulate those with other.
It sounds like what you're doing with your heat study, though, is you're eliminating stuff, whereas with the smoke extract, wouldn't that stimulate stuff?
It's possible that it could, yes.
You might have already answered this, and I just didn't understand what you're talking about. But what was the physical depth that you were taking soil samples from?
I did not cover that, but it was 10 centimeters. So we don't go very deep. And neither does heat. As it turns out that the one experiment that I read about that I got the fire curve or the heating curve from was one where they piled fuel up 9 meters high and 6 meters across, so they could burn that. And with that much fuel, they could get it to go down 30 centimeters to a killing depth. But normally, killing depth on soil is highly variable across a forest area and is very dependent on how much fuel sits there and burns. It's not the heat of the canopy burning, but it's the fuel on the forest floor that, you know, and whether there's coals and stuff that make it penetrate. So 10 centimeters is probably captures it and gets below it, but we'll find out if we get around to the prescribed fires that we want to do.
Did you look at foliar fungal endophytes in the pre-burn forest?
So the question was whether I've looked at the foliar endophytes, and the answer is not yet. But 1 month from now, we're going to go up and do foliar endophytes in the Gaylor Basin up in Yosemite, where we've been doing some tree island stuff with the ectos, and that'll be the first endophyte work of my